everybody. Actually, we had started this chapter 20 is DNA technology and the genomics, and we passed through lots of subjects. Let's just remember some issues we talk about. And the first thing is understanding and manipulating the genomes, and we talk about this one. Actually, we talk about the microarrays, and we talk about the DNA cloning permits production of multiple copies of the specific gene or other DNA segment. We talk about this issue as well, DNA cloning and its application. And we little bit talk again, and we did a preview about this issue and how we can manipulate the gene and how can we transfer the gene and which techniques we would use about this one. Using restriction enzymes to make the recombinant DNA, and we talk about the recombinant DNA technologies and how can we use the restriction enzymes and what are the restriction enzymes and how can we use and how can we get some benefit from the restriction enzymes, etc. And the other enzymes involved in the recombinant DNA technologies like DNA ligase, and this enzyme actually is very important in the DNA replication as well. And the cloning a eukaryotic gene to a bacterial plasmid, and this is a technique, and we talk about this issue. Then identifying the clones carrying a gene of the interest, and we have to find out the desired genes to transfer to another organism. And how can we do? We just explained in our previous course. And these are the techniques and storing the genes and making the complementary DNA or cDNA technologies and cloning and expressing the eukaryotic genes, how to measure and how to detect these kind of things. We talk about bacterial expression system and eukaryotic cloning and expression system as well, and amplifying the DNA in PCR. We all talk about these techniques, especially now, as you're aware, the most of the precise technique to detect if someone carrying the coronavirus RNA, and we are using these techniques. It's about 25 to 100% precision. And we talk about gel electrophoresis as well to separate the DNA fragments, which we digest by using the restriction enzymes, and some other techniques, actually southern blotting, etc. I do remember we talk about the restriction fragment length polymorphism, or RFLPs, and we actually use even in our lab in pharmacogenetic studies, because some people are getting some resistance to the drugs, and we are just looking at some specific genes if they have any polymorphism in their genes. If they have, they are really more resistant, and we are just reminding or let's say suggesting the doctor to change the prescription of the drug. And we talk about the genome analysis, and actually I told you we have some conventional sequencing of the whole genome, but now we are using the very precise and very quick DNA sequence, like new generation sequencing systems, and we all talk about this one, and identifying the protein coding genes in the DNA sequence. It is really important if you know the proteins, because as you know, proteins are carrying some information, and you can get the information from the protein, and you can just make a synthetic DNA by using DNA synthesizers. Then you can prepare a synthetic gene if you wish to transfer to any other organisms. And we talk about this one, and the current estimate about the human genome. We have around 22,000 now. It is a little bit older version of the book. And the determining of the gene function is another important thing, actually. And we have to know which genes are producing which kind of response in the cell stimulation, and this is really one of the important things. And we have now 22,000 genes, but if you think about our leukocytes, they have the ability to produce 10 over 10 different antibodies from the 22,000 genes. And you know, the functioning of the genes is very important, and we have to understand which genes are responsible from which kind of proteins or which kind of RNAs, etc. And this is another techniques we are using now, and this is actually, I think we stop in this slide, and the future direction of the genomics is very important, because believe in me, and in next 50 years, the genomics and DNA technology is going to be one of the most powerful area in all life science, because as you know, every life science, it could be medicine, could be pharmacy, could be agriculture, could be basic biological sciences, and the other sciences must combine or must merge their knowledge with the molecular genetics and the genomics. And the second one for the next 50 years is the most important one, is the artificial intelligence. And by using the artificial intelligence, actually a new era started or launched. It is called as bioinformatics. And you know, from one side genomic studies, from other side the artificial intelligence, and when we combine both of them, and we are getting the bioinformatics, and bioinformatic studies is going to be most powerful studies in very near future. Just remember in the China, you know, when the coronavirus outbreaks and people started, you know, they did the test. If the test is positive, and they took their face and they recorded in the artificial intelligence or the algorithms, and they follow all the people in all the city if they are going outside or they are keeping themselves inside of the house. And this is really one of the big helpful era in the life science, in the society, in the outbreaks as well. And genomics, actually, one of the most part of the genomics is the study of the entire genomes of the every organisms, actually. And now we know the whole genomic structure of the human beings. And before the human beings, we did lots of studies, and we choose model organisms from prokaryotes like E. coli and Streptomyces as actinomycet. And because you know, the E. coli has the circular chromosome, but actinomycetes, they don't have the circular chromosome, and their chromosomes are open-ended and linear chromosomes, and we use the actinomycetes because our chromosomes are open-ended in a linear DNA, and we use these organisms to understand how the genes are working. And this is really good. We get really good information from the actinomycetes and the yeast, and from the plant side, Arabidopsis, and some other kind of the plants are chosen as model organism to analyze their genomes and to understand how the genes are working and how they are expressing themselves. If we only talk about the genomics, it's actually gives some clues, but we have to have a look at the product of the genes, and it is mainly proteins. And the proteomics, actually omics technologies, genomics, proteomics, and now people are talking about lipidomics, and only related with the lipids. And when we combine all of them, genomics, proteomics, lipidomics, then we are now talking about the metabolomics. And all the omics technologies are just derived from the genetical studies, and how the genes are working and how they are affecting whole metabolism and whole anabolism and the catabolism as well. The other thing is single nucleotide polymorphism, or SNPs. It's a short way, and SNPs are very important in our life because almost let's say 2 in 1,000 of our genes are making our differences in all over the world, and the others or rest of them are the same. And the differences just due to single nucleotide polymorphisms, and these are very important. And actually SNPs are the mutations, but if these mutations occurs more than 1% of the population, it is called as polymorphism. And these polymorphic structures of our genes actually are affecting our behaviors and our metabolic activities against the environmental changes and our nutrition and the other kind of things. And the practical application of the DNA technology actually affect our life in many ways, and a number of fields are benefiting from the DNA technology and the genetic engineering. And let's have a look what are they. And firstly we will talk about the medical application. And one obvious benefit of the DNA technology is the identification of human genes whose mutation plays a role in genetic diseases. Lots of things. Actually, we are now talking about the rare diseases, and 85% of the rare diseases are coming from our genes and the mutations in our genes, and we have to have a look at them. But the thing is, these rare diseases are affecting very less amount of the people, so the medical scientists are not studying or are not focusing on these kind of the illnesses, but these are really affecting our lives. And we have to have a look at which kind of mutations in which kind of the genes, and we have to have a look, and we have to find out, then we could then solve these problems with the gene therapy, for example. And even we are just using in medicine in diagnosing the diseases as well. And medical scientists, for example, can now diagnose hundreds of human genetics disorders, and by using PCR and primers corresponding to the clone disease genes, then sequencing the amplified product to look for the disease causing the mutation. When we look at and when we find out which kind of mutation occurred on the genes, then we can just design a protocol to treat this kind of diseases. Even when a disease gene has not yet been cloned, the presence of an abnormal allele can be diagnosed with reasonable accuracy if a closely linked RFLP marker has been found. Then these RFLPs are helping us, and this is actually because of the, again, single nucleotide polymorphisms. Now we are talking about the gene therapy in the medicinal side, and you know, the genes are broken down, or some mutation, some lethal mutation or some important mutation happens in the genes. The expression type of the genes are not working properly, so we have to change the broken genes, and we could help the patients to solve their problems. Actually, we are using various vectors or various techniques to transfer the normal gene to the broken gene, and we are using a kind of different therapy techniques, and one of them is gene therapy using retroviruses. Actually, retroviruses, as you know, we can call them as you know, synthetic viruses, and this is the retrovirus in here, and this is the capsid, and it has the viral RNA. And if we want to clone a gene to the sick cells or sick tissues, we are adding the normal gene to the genome of the virus, then we are sending it to the cell. And we are just sending these genes, and the viral DNA carrying the normal allele inserts in the chromosomes. Then we are just growing these cells in the cell culturing system, then injecting the bone marrow. Then you know, the sick people are getting healthy, and they are getting rid of these kind of problems. And this is one of the techniques in the gene therapy. Now, as you know, people are using the immunotherapy, for example. In immunotherapy techniques, we are getting the leukocytes, for example, in cancer treatment is very common now. In all over the world, these techniques is used. We are getting the leukocytes from the cancerous tissue, and just purifying the leukocytes, and because leukocytes are fighting with the cancer tumors, and you know, in fact, in our body, every day in healthy people, 1 million cancer cells are formed. But our immune system, by the leukocytes, these cells are killed. The problem is sometimes these leukocytes, the cancer fighting cells, are just passing through the cancer cells because there are some defect on their receptors. In this case, in immunotherapy, we are taking the leukocytes and we are just regenerating the receptor producing genes, and we are growing them and in the cell culturing system, purifying them and giving to the sick people. Then the leukocytes are starting to kill the cancer cells again, and this is really promising technique we are using. And we actually DNA technology is used in the pharmaceutical area as well. For example, we are producing vaccines in pharmaceutical area, and sometimes we are purging or pushing some prokaryotic systems or some eukaryotic systems to produce our hormones and our genes as well. For example, as you know, the insulin hormone for the diabetic people are produced by E. coli now, because we cloned insulin genes to the E. coli, and E. coli is producing our hormones. Actually, before these biotechnological studies, people were getting the insulin from the slaughterhouse, and they were collecting the pancreas of the slaughtered animals, and they were purifying the insulin. And these insulin were mixed actually, and belongs to the cow, sheep, goat, pig, and these kind of things. But when we clone the insulin gene to the E. coli, we are just cloning the human gene, and E. coli is producing pure human insulin, not the other animals' insulin now. You know, our big problem now producing the vaccine against the coronavirus at this moment, and people are really working very hard, and you know, it is because of the DNA recombination technologies. Actually, forensic science is using the DNA, and especially we call them DNA fingerprints. And the DNA fingerprints, actually, it depends on the short tandem repeats in our genes, and we are just using the restriction enzymes and just getting the short tandem repeats or STRs. Then we are putting on the gel electrophoresis and looking at their sizes and their repeat times. Then we can find out real guilty, and sometimes you know, people are not condemned the crime, but there wouldn't be any clue. Then they were just judged and jailed for tens of the years. But now we are using the DNA technology, and by using you know, if real guilty people are leaving any evidence like hair parts or you know, anything, and from this evidence we are isolating the DNA and multiplying them, then looking at the SDRs, and then we are just comparing with the you know, the guilty or non-guilty people, and we are really finding out real guilties. And even we are using the DNA fingerprinting in the establishing the paternity, because these are really happens. Sometimes people are rejecting the child, you know, it is not my daughter or not my son or not my children. Then you know, they are subjected to DNA analysis, and by using these analyses, real father and mother are you know, clarified. And genetic engineering actually again used in the environmental cleaning up, because you know, one of my PhD students, she did a PhD thesis, and we isolated a bacteria, actually it's archaea, from the salt lake of Turkey, and you know, these bacteria or these archaea is leaving 2.5 molar sodium chloride solution, and you know, when the polyaromatic hydrocarbons are spread around on the soil, and you know, because of the petro products, we have to use to clean the soil up, and for this polyaromatic hydrocarbon degrading organisms are very sensible to the salt concentration. Then we have to use these organisms to clean up the environment by degrading the polyaromatic hydrocarbons. And for this, the gene salt resistant genes should be transferred to other organisms, and they then become salt resistant, and these organisms can degrade the polyaromatic hydrocarbons. For example, this is only one example, and DNA technology is used in the agricultural application as well. Actually, you know, when we talk about these kind of things, people are mainly thinking about the genetically modified organisms, but genetically modified organisms are not only the plants, and there are lots of different kinds of the genetically modified organisms, but by using the DNA technology, we can produce drug pesticide resistance and insect resistant plants, and we can get much more crop from the plants actually. And we are using the transgenic animals, and these by using these animals we are getting lots of things. If you look at here, and these sheep are pharmaceutical factories actually. I just remind you, I think I told you before, you know, the dolly cloned a sheep, and it wasn't the main project. And the main project of the producing the dolly was to produce the interferon in the sheep milk. And they just injected the interferon gene to the sheep's mammary gland. Then they started producing the interferons for us, because interferon are very important proteins against the viruses, because I think one of the most powerful weapon for human being against the viruses. Then these animals, transgenic animals, are producing this kind of things. Actually lots of new kind of the vegetables and the fruits are even produced by genetic engineering and by recombination the DNA, and we are adding desired genes to get the desired phenotype in the plants. And these are the techniques. Actually, there are some safety and ethical questions raised when we do DNA technology to produce new crops, but I think we have to overcome these kind of problems. And people are saying that you know, genetically modified organisms can spread their genes by their pollen to the environment, etc. But I think the increasing population in all over the world is purge us to produce the genetically modified organisms. And thank you very much for your attention. And I sent the schedule, weekly schedule by CITICS. Please check the CITICS and be ready on the time to follow our other lectures. Could be by using LMS or by using Zoom software. And please have a nice time and be healthy, and see you on next week. Thank you very much.